Hello, everybody. Hi. How's everybody doing? Yeah? So, quick show of hands. How many people are traveling from the Bronx? All right, that's cool. How about Brooklyn? All right. Queens? All right. Manhattan? All right, all right, and Staten Island. I almost forgot poor Staten Island. Yeah. So we have representation from all 5 boroughs. This is great. How many people here have come to Rockefeller campus before? Yes. Oh, my God. So you're all repeat customers. This is great. Do you remember at all what this campus used to be before it was Rockefeller University? Who knows? What kind of farm? Sheep farm. That's right. Just a little bit of trivia, right? in case that's ever relevant in your life moving forward. I don't know. Sometimes you go to like trivia nights at like in some like places. I don't know if they have New York City based trivia. All right. So my name is Jeannie Garbarino. I'm the director of RockEDU Science Outreach, and I'm really thrilled that you guys are all here to share in some pretty awesome science. And so before we go into the talk, I just want to tell you a little bit about RockEDU. Firstly, you can find us on various forms of social media, including Snapchat. Although we don't have our own filter, we're working on it. They keep on denying us for our own filter, but we're getting there. But if you want to put us on or follow us on Snapchat, you can. Or if you want to find us on Instagram, I don't know if anybody even uses Facebook, but we're RockEDU_, also on Twitter. And our hashtag is RockEDU. Additionally, we have a lot of programs that you can return to after you participate in this. So if you're interested, we always have the LOL series on every 3rd Friday of the month. And our next one will be in December on failure. So we talk about science in some of the sessions, but in other sessions we talk about like things related to science, and failure is one of them. And so that's a pretty good thing to talk about, and we'd love for you to continue your amazing attendance streak. Additionally, we have applications open for 2 research programs. The first one I want to tell you about is Lab Jumpstart. Okay, this is a program that goes from March all the way through August. Okay, and so that is a free program. You get a stipend for both the March, the sort of the after-school portion of it, as well as the summer portion of it. But you have to commit through, for the entire time, and it's 21 weeks. So it's a really good program. It helps to get everybody who hasn't had any research experience. It gives you the fundamentals of doing science research, and then you're paired with a research mentor in the summertime to do a 7-week project. Okay, and so this is a really great program if you are interested in getting your feet wet and really learning how to do research. Our second program is a summer program called the SSRP, or Summer Science Research Program. Those applications are also open. The difference is you can just apply to this one, whereas in the Jumpstart program, you need to be nominated by a teacher before you get your application. Okay, any questions on any of that stuff? Okay, but that's good. So if you do have questions, please come see me. I'll be outside after this. A couple of other housekeeping notes. It would be really great if everybody finishes their snacks in the crinkling of their bags before our speaker comes on, because it can be a little distracting. So feel free to just like quickly get your bags opened, your sodas opened, or whatever you have going on, and try to be a bit quiet while the speaker is going, so that it's not crinkle, crinkle, crinkle the whole time. Additionally, after this, we're going to have a series of networking activities that you can come out and participate in. And after the talk, we will ask you to give us some questions, but we're going to make sure that you raise your hand and get the mic. So if you've been here before for an LOL, you know the drill, right? And yeah, so that's it. So now I have the pleasure of introducing Dr. Mark Stoeckle, who actually does a lot of this research in the RockEDU Science Outreach Laboratory. And so we're really pleased that he's headlining this show tonight, because he's such a close collaborator of RockEDU. and an awesome supporter of high school student research. So I'm going to hand it over to you, Mark. Thanks a million for being here, and yeah, we're looking forward to hearing you.
Thank you, Jeannie. It's a great turnout Friday afternoon. I'm going to tell you about some amazing projects that I've had the opportunity to do with high school students. And in these projects, you'll see they got a chance to make scientific discoveries. They attracted a lot of media attention. The media loves it when high school students are doing science, and they changed the world. So, and my hope is that you'll get some ideas from this, and when you go out, you'll think of projects of your own that you might do. So what we do is kind of a combination of the Nature Channel and CSI. I don't know if you watch any of those shows. So we're interested in nature, finding out what's going on there, but we use the DNA methods that are the same crimes that are used at crime scenes. So we work with little bits of DNA, traces of DNA, and we use that to better understand nature. And all of our work is based in New York City. And we've had an amazing series, a run of projects that have turned up some really surprising discoveries. I got started in this actually when my daughter was in high school. I got started working with high school students. She was in high school. I was working on something called DNA barcoding, which some of you may have heard of. It's a way of identifying species by DNA. You don't need to have the whole organism, just a little bit to identify it. And she said, well, can we identify sushi with DNA? And I said, yeah, I think so. I don't think anyone's done that. That'd be an interesting question. So they went out. This is their project. They went shopping, and they liked to eat sushi. They took little bits of them that you only need a little bit like that to get a DNA analysis, and I helped them package that up and send it off to a laboratory. You have to, of course, keep your samples straight, keep your records good. We sent it out for DNA sequencing, got the sequences back, and then we matched these to the database, just like you find DNA at a crime scene. It's only good if the person whose DNA it is in the database. And so what they found was a quarter of the samples were mislabeled, and it was always mislabeled as something better, something more desirable or more expensive. It was pretty much everywhere. There's 6 of 10 grocery or fish markets and 2 or 3 restaurants. And for instance, something sold as white tuna was really Mozambique tilapia. So a very expensive fish, the cheap fish sold as an expensive fish. This attracted a lot of attention. They were, Kate and Louisa's story was on the front page of the New York Times. They were on morning television, and they were in newspapers really around the world. This is a cartoon from the Korean Daily News. I think you can see there are 2 girls looking at a fish on a plate with a magnifying glass, and the chef being led away in handcuffs. And that's sort of the idea, that something wrong with the fish that they discovered. And without the student involvement, it absolutely would not have gotten the attention that it got. And this did appear in scientific literature, but it was the popular literature that really made it happen, meaning the Times and the television, radio. This is a quote from Time magazine. If you're paying for white tuna and you're eating tilapia, I think you'd want to know that. That was a summary of that. Their story is now in college textbooks, and they have their picture in college textbooks. So again, a lot of impact from that. The samples were, and their results were part of a scientific paper that was in Food Research International. And this study, because it got a lot of attention, it really helped make other changes. And one of them was, it led to much bigger studies. This is a study that Oceana, which is a nonprofit group, did across the country, hundreds of samples, and they found really the same finding. About a third of seafood is mislabeled. And these things prompted the Food and Drug Administration to adopt DNA barcoding as their official standard for identifying fish. So they went from high school students to national exposure and international exposure and really having a lasting impact. This was such a success, we did it, went again the next year, we did a project called DNA House, and we asked Brenda Tan and Matt Cost to just find anything you could find in your house that might have DNA in it. You know, how good is DNA? And they tested, they had amazing results. Things like if you get a feather duster and it's made from ostrich feathers, you can get ostrich DNA out of that. If a dog biscuit that just, you can analyze that and you can find out what kind of meat the dog biscuit is made from. So you really don't have to have a piece of the original animal. They did test some food items, and that got picked up by of course they found things that were mislabeled. Something that was labeled as sheep's milk cheese was really just cow's milk, and something labeled sturgeon caviar, this is from a very high-end delicatessen in Manhattan, was really from Mississippi paddlefish, which is good to eat, but it's just not sturgeon. And here's it says, you can see 2 high school students were part of this study. They were in the New York Post. It's hard to get into the New York Post unless you've really done something wrong. But this was, one of the things they found was that they tested some cockroaches or pieces of cockroaches. There were, I won't say where we found them, but they were genetically distinct from any cockroach in the database, and it seemed like it might possibly be a new species, and that intrigued the New York Post. That was such a success, we tried again, and this time looking not so much at the animals, but at plants, and Grace Young and Catherine Gamble and Rohan Kirpikar went around, got tea bags, or loose tea from the school cafeteria and from stores in the neighborhood, and they got regular tea, which is made from the tea plant, as well as herbal teas, which have a variety of things in them. And we got the testing done at New York Botanical Garden by Damon Little, and they found, particularly with the herbal teas, 1/3 of the herbal teas have things in them that aren't on the label. Now, sometimes it turned out it was things like chamomile, which is a good thing to put in tea, but it was a major ingredient, but it's not on the label. And then there are a lot of things like lawn grass and a whole variety of weeds that are in the herbal teas. So their work is a basis. This is a scientific study that was in Nature Scientific Reports. You can see their co-authors on that. They were also in the New York Times. And that study led to a much larger study done up in Canada. You can see the tea study was the first reference from that. And this really annoyed the people that make herbal products because they said, you're giving us a bad name. But the fact is that a lot of them are mislabeled. And this was really helped bring that to the fore. And this led the New York State Attorney General to sue the herbal manufacturers for, and since then the verification process has changed. It's obviously in the interest, if you're selling an herbal product, to know what you're selling and to be able to authenticate it. So that really had a big impact. We did something a little different for the next project. This is working with Joyce Zio, as a senior at Hunter High School. We wanted to follow up on that cockroach story. And it turns out there's not a lot of research on cockroaches, but no one wants to even pick them up or go near them. But what we did was we wanted to find out, are there really different species in New York of the really large cockroaches? These are called American cockroaches. And we set up a website, and we emailed everyone we knew, and we asked them to mail us cockroaches, not alive. And we had to check first, because it turns out it's okay to put a cockroach in the mail because it's not officially a pest, because it's everywhere, so it doesn't matter whether you spread it around. And it's not considered wildlife. If it's wildlife, then it's under the protection of the U.S. Fish and Wildlife. So it's all right to put a cockroach in the mail. And for the next year, every time, these are all letters that contain cockroaches. So I would come into work and open the mail, and we had a wonderful selection of cockroaches from all around New York City, from other parts of the United States. And people heard about this. We weren't trying to, but we got some from Argentina and Australia, Korea and China, from Iran. So that was a great, and the project was, it's 4 or 5 years ago, and people are still mailing me cockroaches. So I have some up in the lab. And they're very disappointed when I tell them the study is closed. I said, well, I'll put it in the freezer. So we have a lot of things in the freezer. It turned out the cockroaches, they're different types, and they're a little bit different by the neighborhood, but they all interbreed. So that's one definition of a species. So they're probably like New Yorkers. So they came here from different parts of the world. And now that they're here, they're mixing and matching. And so this was the scientific study. You can see Joyce was a co-author on that, and said interbreeding among these deeply divergent lineages, these genetically distinct forms. We had some wonderful publicity. This was in Wired magazine. What's it like opening your mail and finding dead roaches? This was Russian television, and they came and filmed it here at Rockefeller, actually right over here in the student lab. And among, not am I still getting cockroaches, but among certain of our friends, this is, every time they tell me, they see me, they think of cockroaches. So it's such an unusual idea. So this is what I'm going to talk about, what we're working on now. And this is a really exciting new technology with DNA. So this is working with, before working with something that's sort of a recognizable specimen, a bit of tissue, a wing of an insect. Here, we're just collecting water, and we're analyzing the little bits of DNA that are floating around in the water. And it turns out, this is work that was started initially in France, that there's enough little bits of DNA in the water that you can identify things that live there just by taking like a glass of water. So the DNA that's floating around is called environmental DNA, meaning it's in the environment, it's not part of a plant or an animal. And these are, does anyone know what fish this is? It's a striped bass. Okay. What's that? Okay. I discovered one thing with this project is the only names of fish that people know, and I was the same with this, are fish that we eat. Well, we do eat striped bass, but there actually turns out there are dozens of different species of fish that live right around New York City. So this is our research site. That's Manhattan, and you can see parts of some of the other Brooklyn and Queens and the Bronx. There are a lot of people that live here, but it turns out there are a lot of fish, and this is actually an important environment for fish. And the harbor is cleaner than it used to be, and one of the results of that is more fish. They've started to come back here, and then the fish attract things that eat fish. So we've been, some of the collecting we do, this is Rockefeller right here, is right outside here in the East River. We started this with Iman Nassef and Alden Liang. Iman may be here, I don't know. And first we went up to Central Park and went to the Harlem Mere, and we collected water up there. We said, this is the first time we're going to try it. I thought it can't possibly work. But it worked amazingly well the first time around. So we got these DNA out of there, and we amplified the DNA so we could look at DNA of vertebrates, which all fish are vertebrates. And then we see, can we match these very short sequences to the library, to a GenBank, which is like our database of criminals. And we got 7 species of fish, And these bars give you an idea how much of the DNA of that fish is in the water. So, and there's a lot of roughly, if there's a lot of DNA of the fish, then there are a lot of the fish. But that's an area where a lot of people are trying to do research with that. We also found other things. Human DNA is actually everywhere we go in the water. I mean, I haven't been out to the middle of the Atlantic, but anywhere along the East Coast, certainly we get human DNA, because there really are a lot of us. Other dogs, you can imagine rats. Rats are the commonest wildlife. We get their DNA in the water if you sample around Manhattan. Some birds, and also raccoon, which spends its time around the water. So that worked amazingly well that first summer. If we compare it to traditional methods like putting a net in the water or in fresh water, they do a thing called electrofishing, which is you have a generator and you put an electrode in the water and you stun the fish. You electrocute them. Hopefully they survive. And that way you can pick them up and count them. So it's sort of a primitive method, but that's still how we survey fish in fresh water. And a nice overlap with what's found either by electrofishing or by seining, by using a net. And we found some things that were not found in the same water by either of those methods. So that was good. So then we said, well, we'll just try some other areas with water. Right out here in the East River, and the East River is not really a river. It's really a tidal channel, and the water flows back and forth between the harbor at the south end and Long Island Sound at the north end. So it's really a big ocean. It makes a sound. Uh, misconception? What it, say all of our food, and somehow there's some idea that DNA shouldn't be there. I'd say that's the biggest misconception, or lack of knowledge, really. And the fact there's enough DNA in the water
Cool. All right.
Down here.
I was wondering how long DNA stays intact in the water.
That's a really good question. We need more experiments on that. I'd say it lasts days to maybe weeks. If it lasted forever, we wouldn't see any difference from one season to the next, and we'd get eventually, the DNA from a fish that lives in Asia would make its way over here, and the ocean would be more like a bowl of soup. But it's kind of a Goldilocks quality. It lasts long enough that we can detect it. As I said, sort of probably in the days, maybe more than a week, but not so long that it's not just not a good record.
Great. Here we go. Over here. So you get a microphone and a water bottle.
What was your motivation to research on eDNA?
I read about it a few years ago. The first paper is done in France, and they took very small samples of water, much more like a quarter of your water bottle, and they used it to show that frogs were living or not living in a pond. And I don't know, I saw that, I thought right away, this is going to be a big thing. Now that paper was 10 years ago, so it takes time for these and the techniques and then to be disseminated. But as I said, I like finding out things that no one else knows. No one else knows. Every time we go out and sample the water, I'm excited to see what we get.
Besides students, who else could benefit from this talk?
From the talk? People that are interested in the environment around New York City. I think there's a lot of general interest in the environment and protecting the environment among adults. And we tend to think the environment is a thing out there, but in fact, we're living in the middle of a natural environment, and we want to do things that can help it.
And just to add, I mean, there are lots of groups that want to use this technology for their own personal surveys. People that are interested in the health of, for instance, the Gowanus Canal, or maybe the Bronx River, or maybe, which is the only freshwater river in all of New York City, by the way. So, I mean, this with high school students, Dr. Stoeckle is actually pioneering a super useful technique that can benefit people at all different levels. So I think, that's really important. Okay, let's see. We have a question. Oops, sorry. Here's
What's your favorite part when you're conducting an experiment?
I guess when you get to the part of it where you open the book and you see what you got. And depending on the experiment, that happens at different times. Sometimes you get a computer file and it has lots of sequences in it, but you don't know what they are. But then when you match it, it's really like, pulling back a cloth and seeing what's underneath. Now, sometimes we get nothing, but that's the favorite part.
All right, I have a question over here. And then I'll get.
In the DNA, like barcoding, right? In the water, did you find like other organisms other than fish?
The way we're analyzing it, we're amplifying the DNA, so increasing the amount of DNA, and we're targeting vertebrates. So there's lots of other DNA in there. There's bacterial, viral, plants, and as well as the invertebrates like crabs. The way we decided to focus on vertebrates, and those things are interesting too, all of those.
And like other waters, other than New York, did you go to like other like states and observe their areas of water? Find like
I mean, there are a lot of groups doing this. Most of it so far in the U.S., just the way things developed, has been done in fresh water. There's a lot of work in the Great Lakes looking for invasive species. There are a lot of species that are already invasive species in the Great Lakes, and there are other ones that are just at the edge. And the fisheries managers there, they use this routinely. In the oceans, it's still new. Not many people, usually to do anything in the ocean, you need a boat. You have to go, and that would be interesting too.
Holy moly. All right, we didn't get anybody from over here yet. Okay, let's go. Here we go.
Do you have any examples of some data that you found very surprising while conducting your research?
You're saying data that's, as I said, well, first of all, it's all surprising to me because it still doesn't quite make sense to me that in a small volume of water there's enough DNA to tell you, a dozen different fish that are in the neighborhood. I was surprised we could find the dolphin DNA. Relative to the number of fish, dolphins are predators. And if you've done any ecology, there are lots of prey animals. And then when you get up top of the pyramid, the biomass of a predator is much smaller. And I was surprised, I was pleased, but I was surprised with that.
How can you use your findings in the future?
How can
you use, based on your findings, how can we use this in the future?
The main way it's going to be used is to understand much better what lives in the ocean. If we talk about fish for a minute, really our knowledge is really quite primitive. If you were to compare to, say, what we know about birds, we have counts of every bird species on the planet, how many individuals there are. We know where they migrate. We monitor them every year. In the ocean, it's like we go like this. We do a test here, we do a test over there. Now, the oceans are bigger, and the reason we do so few tests, and then we try to extrapolate from that, is because it's so expensive. It's $10,000 a day to put a boat in the water with a net in the ocean. If we can do it through water samples, and eventually it's going to be autonomous, it's going to be undersea robots that are going to be out there collecting the DNA or sitting on the bottom and maybe analyzing it there and sending a signal to the surface.
With different DNA in the water, is there anything you might have seen in the DNA besides the species?
I'm not sure a good answer to that question. I mean, other than we are just looking at fish, it's not enough. Some people say, well, is the DNA damaged, from the environment or mutagens, carcinogens in the environment? We couldn't tell if that was true or not because the pieces we're looking at are so small that you would really, and they may have been damaged just by being outside of the animal. But that might be an interesting question. But for that, you'd have to look at the animals themselves.
All right, so we had someone who's been raising their hand for the entire time. So here you go.
How would future climate change affect the DNA in the water?
I think everyone who works on the environment or ecology is concerned about climate change. We don't know what exactly is going to happen. This gives us a way of monitoring what's happening. As I said, we just don't know much about fish and fish populations, but we don't. But now, if we can do this kind of thing, and we do it every year, and we collect the data just like you measure the temperature, like you do a weather report, but you have now, and they collect temperature from many different weather stations. So if we can collect DNA information over the course of a whole year from many different states, we'll be able to see are species moving north, are they disappearing, are they changing their routes? So I think as a telling what climate change is doing to the animals, it can help us.
All right, so here, you've had your hand up, but we're out of water bottles, so yeah.
Still ask their questions.
Okay, so I know you're only looking at vertebrates for now, but can environmental DNA also be collected outside of water?
Yes, you can. I mean, water is a convenient way to get it. You can do from the air. There's DNA in the air. It's a little, not a lot of it, but that's one thing you could do. And from soil, those would be the main places it's been done. So those would be all alternatives. You don't get like a stream kind of integrates the DNA from the whole distribution, the whole watershed of the stream. And when you're doing a soil sample, you probably don't get that. You're getting a much more localized answer, but it's going to be an interesting answer.
If I can add to that, so there have been studies done on the subway where they basically looked at the microbial makeup of every single subway stop. And they also did air samples with looking for DNA. And you know how sometimes when the train comes and you feel a little breeze right before the train comes? Yeah, well, in that breeze, I'm really sorry, I'm going to gross you out right now. Do I or don't I? Do I or don't I? Yeah, okay. In that breeze is DNA that originates from rodent feces. Welcome to New York City, everybody. Yeah. All right, next question. We have someone over here. All right, settle down. I'm sorry that I riled you up here. All right, let's hear it up for the next question, okay?
How have you communicated your findings with other scientists?
I'm sorry, could you say it again?
How have you communicated your findings with other scientists?
That's really important. I mean, that's kind of half of science. It's not just doing it, but it's communicating it. I mean, there's several ways. One is publishing a scientific paper and then trying to publicize the fact that you did a scientific paper. For each of these projects, we've also invested a lot of effort into getting publicity, not scientific publicity, but getting publicity in the mainstream press. Because in the end, the reason we do science or we're allowed to do science is because of support from the public. So I think communicating those findings to the public is in as wide a way as you can. And this is information that's understandable. So that's another way. And then the third, I'd say, is local networking. We have a discussion group of researchers and funding agencies from around the New York, New Jersey area to find out. There are a lot of people concerned about the health of the Hudson. What can we do, or how does our information help them?
Question right here.
You said that the DNA lasts only for a few days or weeks. What happens to it after that?
Excuse me, what happens to it?
Yeah.
Well, it gets smaller and smaller, and and you're, it just gets shorter and shorter, and that means harder and harder to detect. The ancient DNA studies where they're getting DNA out of like soil samples or sometimes bone, but sometimes they can get DNA bits that are 50,000 years old. That's very short. Those are 10 or 20 nucleotides long. This piece is about 200. So it just gets shorter and shorter, and then eventually it's completely broken down into the sort of chemical constituents.
Okay, we have a question up here in the corner.
What type of difficulties did you face while doing this experiment?
I'm sorry, what type of?
Difficulties did you find while doing this type of experiment?
Difficulties. Well, first of all, collecting the water, to do that whole year, that looks like some of the days it was really bad weather. And just to go down to think, oh my God, I'm going to go down to the East River, I'm going to go out to Coney Island. I've been out to Coney Island when you put a bucket over the side of the pier and it just blows, you know, straight out. I had to put weights on it to get it to go into the water. That's fun in its own way. The main part of, there's a part of lab work that it's very meticulous. And, you know, you're doing, like working with these tiny little volumes, and you have to do it over and over, and you have to not make a mistake. So that's a hard part.
I have a question down here.
Okay, so this might be complicated, but like, exactly how many species did you find, like, throughout all of your like DNA findings? Like,
for fish, about 100. Okay. So that includes the samples from South Jersey, as well as the East River and the other samples that were there. And we have, out of that, I'd say 20%. When I say 100, I know it's a distinct DNA, but I don't actually know what the species is. I know it's some kind of ray, or it's, but it's not in the database.
All right, so we'll take 2 more questions. You'll have to fight it out. Rocks, paper, scissors. Sorry.
Sorry. What kind of limitations are there to using your method compared to electrofishing and seining?
The biggest limitation is we don't get any information on the age or the size of the fish. So is the DNA coming, it could even, you know, from a little larval fish, and there are just lots of them, or is it coming from, you know, very large adults? That's things that if you're trying to assess the health of a population, you'd like to know. So that's an inherent limitation in this, this approach.
Last question. Oh, we have one over here. Okay, here you go.
So about how is this eDNA sampling technique applicable to like C4 sampling, or like more oriented on the the microbiome instead of like more?
I'm sorry, can you say again?
Okay, so is this eDNA sampling technique more oriented towards like a macro scale DNA, like macro scale organism DNA sampling, or and like categorization, or is it also applicable to like more, you know, bacteria, stuff like that in the water, and how that's distributed?
Yeah, that's a good question. It's the procedure we follow is really adapted from microbiome studies, where they're looking at bacterial diversity, and we took that same approach and said, okay, we're going to look at vertebrate diversity. So same approach.
All right, so let's give a hand to Dr. Stoeckle. So I'd love it for you guys to just hang out outside for a little bit. We have a couple of cool activities, some true-false trivia questions on DNA. And yeah, so thanks. And don't forget to look us up, rockefeller.edu/outreach, and apply.